Sorry to put you on the spot.
Hello, everyone. We're going to get started for today's Tuesday lunch seminar. It's my pleasure to introduce Aldo Arellano. He recently moved to Ann Arbor after getting his PhD at the University of Wisconsin. So he's going to be telling us about his PhD work. Here in Ann Arbor, he is a postdoc with Greg Dick at Sigler, which means it's been an interesting time to start a federally funded postdoc. Yeah. But he's very much an EEB type of person. So I'm really excited to have him here to give this lunch seminar.
Awesome. Can you all hear me all right? Is that working? Okay, cool. Yeah, so thanks thanks everyone for coming out today. Happy to get a chance to introduce myself to the EEB community. As Meg said, I just started it with Sigler. It's been a complicated start to say the least. But so today I'm going to be talking about some of my dissertation work on the pitcher plant mosquito and its environmentally acquired microbiome. It's doing nothing. Okay. Oh, yeah. But I want to start by sort of introducing just a bird's eye view of the kinds of questions that I'm really interested in, regardless of the system. And these have to do with community ecology spanning multiple scales, so bridging connections between macroscopic and microscopic scales. In particular, this is thinking about community dynamics and how they're linked at both of these different scales of biological organization. So it's a common thread in all the work that I've done up until this point, whether this was as an undergrad working on cyanobacteria blooms in Lake Mevat, which experiences huge emergence events of these midges, working as a technician, exploring cyanobacteria heterotroph interactions, linking that to global nitrogen cycles, or recently in my PhD, working on the aquatic microbial communities that are housed by pitcher plants, how they're acquired, curated by the pitcher plant mosquito. But obviously, it's a huge challenge to tackle multi-trophic systems, multi-scale systems, so we need tractable models to do that. And the importance of multi-scale thinking, I think, is highlighted well in this recent review, where the authors considered how food web nestedness and dynamics at 3 different scales, so the microbiome scale, the, oh, I need to press OK on this, sorry. The microbiome scale, the host scale, and the multi-host population scale are sort of interconnected. So dynamics in one can cascade out into others. Another motivating theme in my research, more recently especially, has been microbial symbiosis across ecological and evolutionary gradients or transitions. We considered this question recently in a recent call to action in the microbiome sciences, where we're using dipteran insects as in particular, dipteran insect vectors as a case study to think about how our understanding of host-microbe interactions is enriched by using non-model organisms. So a lot of our insights into host-microbe interactions is derived from these lab rat models that we keep from multiple generations in the lab under controlled conditions, but we can learn a lot from broadening that scope to encompass the natural diversity in related hosts, just off flies as an example, dipterans, true flies as an example. So this brings me to my study system of my PhD, which I think is the perfect system for this job of multi-scale exploration, as well as using an interesting comparative model. So this tripartite or 3-part symbiosis is between the purple pitcher plant, the pitcher plant mosquito, and their associated microbiota. The pitcher plant and its obligate mosquito symbiont, they occur throughout this range natively, as far north as parts of Canada and as far south some populations in northern Florida. The pitcher plant is carnivorous, so it produces these specialized leaf structures called phytotelmata, or pitchers, which I'll refer to them as pitchers throughout, that have these downward-facing hairs, where insects landing upon these hairs are forced to fall into these waiting rainwater that is collected in these pitchers, where they drown and are degraded. But the way in which they are degraded is pretty unique in Sarracenia, this genus of pitcher plant, because it doesn't encode a lot of the necessary enzymes to actually break down any of those insect exoskeletons. even though it relies on carnivory as a way to subsidize its nitrogen requirements in these naturally waterlogged and nitrogen-depleted bog soils where it occurs. So a lot of this activity is outsourced to the microbial community. In particular, you'll note that chitinases are not endogenously encoded by Sarracenia, chitin being a main component of insect cell wall, exoskeletons. So that's sort of a problem. And that's, again, outsourced to the pitcher-associated microbiota. Wyeomyia smithii, which is my focal insect and model for this talk and my dissertation work, is an obligate symbiont of pitcher plants. It develops exclusively inside these water-filled pitchers. So it progresses through 4 immature larval instars, or immature stages, as well as, which are all aquatic in the water-filled container, as well as an aquatic pupal stage, before emerging as a terrestrial adult. They're holometabolous insects. They undergo complete metamorphosis, like how a butterfly would undergo progression from egg to caterpillar to chrysalis to adult. It's also unique because unlike other mosquito species, which you might think of as laying eggs in any old body of water, and thus coming into contact with highly variable microbial communities, a female Wyeomyia smithii only lay their eggs inside of these water-filled pitchers, meaning that they are consistently being associated with at least relatively more simple microbial consortia that's found inside of these pitchers relative to your dishes under your potted plants, overturned tires, these sorts of things that other mosquitoes are known to oviposit into or lay eggs into. So this is partially why in most mosquito systems, we don't think of mosquitoes as a host that has a dedicated microbiome per se. It's very transiently associated, it's non-specific, but in a case like this, there's a higher possibility of more consistent interactions, perhaps more co-evolved interactions to emerge, given this frequency of interactions with a particular consortia. Wyeomyia is also a really rich comparative model to use in the study of insect-microbe symbiosis in general. For other aspects of its unique ecology, as if being a symbiont wasn't unique enough across mosquitoes, it also has this cool blood feeding behavior where only in the northern part of its range does it blood feed. Sorry, not blood feed. And in the south, it does blood feed, or occasionally will blood feed. And this is probably in opposition to what people typically think of for mosquitoes, which are blood feeding, because the females at least, because they need to take a blood meal in order to provision their eggs. But there's actually many lineages of mosquitoes, and here I'm just showing on the left a simplified phylogeny, wherein there are populations or species that never have to take a blood meal at all. Wyeomyia smithii being the focus here. And this trait is known as autogeny. So if the species is autogenous, it's non-blood feeding, and autogenous meaning it is blood feeding. And we know that from other model species that host physiology during blood feeding is affected by particular host microbe interactions. And in every species we've looked at so far, mosquitoes require a living gut microbiota in order to develop. So in the absence of a microbiota, they will arrest at the first larval instar and not progress further. And this is mediated by, we think, light-sensitive B vitamins, which are constitutively released by the microbiota under normal circumstances in the field. So exploring host microbe interactions over gradients of life history and blood feeding can give us insights into what are conserved features of host microbe interactions and what are unique features of host microbe interactions, given those transitions evolutionarily. And of course, I have a bias for Wyeomyia, but I'm showing just a smattering of other cool mosquitoes that I think you should all go look up and look into, because they have really cool ecology. There's over 3,500 documented species of mosquitoes, which span multiple life histories and biologies. From Sabbathes, which have that iridescent blue one on the top left that has these paddles that they use in in courtship displays. Uranotaenia, which are obligate feeders on leeches and other invertebrates. Malaya mosquitoes and Toxorhynchites mosquitoes, which are also non-blood feeding. Malaya has this modified proboscis that it uses to trick ants into regurgitating honeydew. So that's what it eats when it's an adult. And then Toxorhynchites, which are predaceous as larvae, so they actually eat other mosquito species. So there's a lot of unique life histories that are emerging in parallel to non-blood feeding, for instance. Today I'll talk about really 4 short takeaways from my dissertation research. So first of which is, what are the trials and tribulations of bringing a new species into the lab in the first place? So bringing Wyeomyia into the lab as this comparative model. Moving out of the lab back into the field, are mosquitoes actually, and these larvae, acting as uh, top-down influences on these ecosystems, these mini ecosystems, micro ecosystems as they're called in the literature, so affecting who is there and what they're doing. And then thinking about if that process could be adaptive or beneficial to mosquito development. And lastly, some mosquitoes go to sleep for the winter in the process called diapause, which is a hibernation-like state. And in some ongoing work, we're thinking about how mosquito-microbe interactions change during diapause. Yeah, so starting with this first story, what we learned when we bring a new species into the lab. I'm not the first person to think about this. Obviously, we wanted to contribute to work in non-model systems, but this involves doing the comparative work of knowing what are we losing if we're trying to domesticate this species in the lab relative to what we'd see in the field. Are the insights that we would glean from laboratory work consistent with what we see out in the wild? So people have thought about this from eagle rays to ring-tailed lemurs, and a lot of the work in my PhD lab, others in the lab worked to develop best practices for recapitulating microbial communities in the lab from those that we find in the field. So we're trying to apply some of those methodologies here and just get a baseline really with this first story I'll tell you about for what are we losing and what is consistent. So the first thing we did was go out and find a field population of these mosquitoes and these plants in the wild. So I was at UW-Madison, just a few hours drive away at Cedarburg Bog outside of UW-Milwaukee. We have a field population in Saukville, Wisconsin that is accessible via this boardwalk. So you see there's this boardwalk over this bog area, and leaning over the side or sometimes getting into the bog, you can access pitcher plants which are found there and have a natural background colonization of Wyeomyia smithii. So Carrie and Erica, shown here, my PhD advisor and one of my committee members, they helped me out in the field where we're profiling bacterial communities in mosquito larvae and their surrounding environment. On the laboratory side, we got Christina Hotzapfel and William Bradshaw at U Oregon, who had kept a colony of Wyeomyia smithii for some time for other reasons related to diapause in culture. And that population is also derived from Wisconsin, funnily enough, and lucky for us, because we had a sort of a little bit, not exactly one-to-one, but more representative laboratory population relative to our field site. And what we did is that we brought those into the lab ourselves, got up our rearing procedures, and profiled bacterial communities over the entire course of mosquito development. So looking to see if there are any inherent dynamics to microbial community assembly from the egg stage through adulthood in our laboratory colony. And just to show some numbers of husbandry, because I feel like husbandry is an unsung sort of difficulty in doing any of these sorts of things. These require daily care. So that was me for most of my PhD, daily going in. Now a new grad student has taken it over, so I don't have to make that commute. And that's about 300,000 mosquitoes that we've counted and circulated for that, just the period of time that I was there. But 0 bites, because this is a northern derived population, so we don't have to worry about blood feeding, which is a kind of an annoying aspect of rearing mosquitoes if you've worked with any other species or talk to someone that usually works with more lab rat species like Aedes aegypti or something. And when I'm saying profiling bacterial communities, the methodology I'm referring to is amplicon sequencing. So we take total genomic DNA, which we extract from water or animals, in our case, the insects, and then we selectively amplify out via PCR a region of interest. In the case of bacteria, the 16S rRNA gene, which is conserved across bacteria, but variable enough over evolutionary time that we can use it as a marker gene to profile what bacteria are present. Or in some other work, we could use ITS to profile fungal communities as well. Yeah. So what do we find in this profiling in comparing our laboratory and our field populations? So I'm showing relative abundance graphs, which all sum to 1. So it's the relative abundance of different bacterial orders in this case, faceted by our different sample types. On the left, field larvae, field water, following that, and then our lab populations, larvae and water. So there are differences markedly that you can already see by eye. But to distill some of the high-level information that I want you to get out of this graph, we can see that what is conserved, at least, is that in larvae, so comparing larvae, so the left on each of the field versus lab facet sets, there's more yellow, right? So we have this enrichment of yellow, which in this case is actinobacteria, and we have relatively low representation of some of those blue colors, which are more common in the environment or their surrounding environment, which are members of the Bacteroidetes. What we might also note is that there's many more colors in those field samples, meaning that we're losing a lot of taxa when we're bringing this species into the lab. And that's consistent with any sort of work that you do in trying to domesticate species. You're bound to lose a lot of what's naturally present in the wild. You're just not provisioning that community with what it needs to persist. And in this case, we don't have the host plant or the natural environmental context. But it is encouraging to see that we have some consistencies in what environmental filtering is happening on the host side. So we also profiled development over time and the composition of the community over time. So we can see that the egg community, larval community, and adult communities are all pretty distinct. So we have dynamic microbiomes, dynamic bacterial composition over time. And some things that are relatively rare in adults, like those are really common in larvae, like those actinobacteria again, whereas we have relatively few Bacteroidetes. So again, those blue colors which bloom in adults. And even just in adulthood, for instance, from newly emerged on the left to having been sugar-fed, we see this massive difference. So you don't have greens anymore, it's all blues, meaning that we have this really diet-dependent microbiome that's also establishing. So we have variation over developmental time and variation just via diet. And again, a lot of these are consistent features in other species, so it was good to see this was internally consistent. Another thing that caught our eye was this low diversity community that was present on the egg surfaces. So we can see, so CNV just refers to conventional. So this is just in our colony, we go and collect a sheet of eggs and we crush that up and extract DNA from it and say what bacteria is there. And it's really dominated by all of these beta proteobacteria in that case. And we have marginally more complex community in our sterilely reared setup. So we just had adults that were derived from, or individuals that were derived from the colony, raised under sterile conditions where there's no exogenous inputs into the system, and then those adults lay eggs in closed containers. So slightly different community. In either case, low diversity. And this, and this got us thinking about what that low diversity community could mean for adults going to inoculate sites when they are laying eggs in the wild. And I'll talk about that a little bit later. Another thing that we wanted to do when bringing the species into the lab is adapt gnotobiotic methods. So we went and said, can we selectively clear and reintroduce bacterial taxa of interest into our system? So this might be familiar from mouse work interrogating like gut microbiome host interactions where you can rear germ-free mice and then selectively inoculate them with defined sets of microbes. And in our case, I'm showing our process on the left where we start with surface sterilized eggs, maintain them on an irradiated diet of, in this case, freeze-dried brine shrimp and guinea pig chow in a 4:1 ratio, which is just the literature standard for their diet, and then any defined microbial backgrounds that we're interested in. And we did this with a handful of field-derived and lab strains, so and as well as in a xenic background, so that is germ-free background. And pleasingly, it's consistent with other systems where axenic individuals fail to develop. So the proportion pupating is 0 out of 120 larvae. So because none progress past the first larval instar. So they just like other mosquitoes, need those microbially derived metabolites in order to induce development. And but there's high variation in the degree to which a given isolate's able to rescue development to the same level as their native microbiota, which is shown all the way on the right. And this is interesting because if I had run this experiment with Aedes aegypti or another blood-feeding species, all of these would have been equivalent. And it's interesting to note that this non-blood feeding species that has this unique life history also has seemingly more specificity in the degree to which any given microbial isolate would recapitulate robust development. So while the feature of microbial dependency is retained, the specificity is higher in the species. And in another facultatively autogenous species, so a different species that sometimes drinks blood, this is something we've also noted. So we haven't studied it sort of rigorously enough to make that direct claim that autogenous or facultatively autogenous species have more specific microbiome requirements, but I'm just noting 2 points and potentially something of interest. All right, so for this first vignette, we found that symbiotic association between Wyeomyia mosquitoes and pitcher plants hasn't changed the dependency that this mosquito has on microbes in order to develop. That's conserved across the phylogeny in all species we've surveyed to date. And further, we see that pitcher plant taxa are able to colonize this mosquito and are likely dispersed by adults, and that particular community members are better than others at sustaining development. So these are our sort of bottom-up effects of microbial community on mosquito development. But we wanted to think about mosquito effects on the community in its natural context. So we saw this evidence again in the lab of bacteria sustaining development, but what about the other way around? For this next project, we went out to the field, back to Cedarburg Bog, to explore top-down effects of mosquito larvae on microbial community composition and function. So mosquitoes are filter feeders. They're feeding on bacteria and microeukaryotes in the environment. So we wanted to see how that behavior is affecting the microbial community composition and what that community is able to do. In particular, thinking about ecosystem function. So what function is it that functions that are essential or dictate essential processes in these microenvironments is being influenced by pitcher plant mosquito colonization and density. So yeah, we went to Cedarburg Bog with Dr. Erica Young and Carrie, and we found newly opened pitchers out in the field that hadn't been visited by mosquitoes yet. And we tagged them and established 3 different treatments, enriched, in which we every 10 days would come and add mosquito larvae. Unmanipulated, we didn't do anything to those, and depleted, where every 10 days we'd come and exhaustively remove any mosquito larvae that were present. And these larvae are coming from our laboratory colony, so we're bringing our germ-free mosquito larvae out to the field to add them to our field manipulated pitchers. And throughout this time, we're also taking samples for microbiome composition. So we have pellets from the water inside of these pitcher plants of our different treatments, about every 10 days for 74 days. And at several time points in the middle, we also took real-time measures of enzymatic activity, so we can correlate intensity of larval colonization with changes in enzyme activity. So this is proteases and chitinases, both of which are essential for the plant to degrade insect prey. So this is that ecosystem function. And then at our last time point, we also took samples to query the resident microbial community at the end of this persistent these persistent treatment effects, to see how they could differentially hydrolyze carbon substrates, so a panel of different types of carbon. We also did micro-incubations to see how active those communities were, and measured macronutrients at the end. So how much carbon is there, how much nitrogen, how much phosphorus, and is that altered in any way by the presence of these mosquitoes at these different levels? And this is over primary succession, I should note, because these pitchers, when they first open, are completely sterile. So we are following the community assembly over time, de novo, I guess you could say. I also just want to shout out some other work that we're working on, and I'm not going to have time to talk about today, but we only sequenced the first and last time points on what we've published so far on this study, but we were able to get some money at UW-Madison to do additional sequencing of some middle time points, and also to do some ITS sequencing. So we're not just thinking about the bacteria, but also the fungi and how bacterial fungal interactions could be affected by higher order trophic processes like mosquito grazing, mosquito activity, and also we're trying to recapitulate some of that in the lab. or not currently anymore. We were trying to recapitulate, and we're working on writing it up right now. And Jelena and Julie are two wonderful undergraduates who helped with a lot of that work. But, and I would be happy to talk about bacterial fungal interactions after my talk. I have a lot of questions about it. So getting back to our manipulation here, we were successfully able to maintain our different levels. So it wasn't just that every time that we went out and manipulated them, they fell back to where they were naturally before. So we had lasting effects of our treatments out in the field. Enriched again, stayed enriched, deplete, stayed deplete in these box plots over time. And jumping into some of what we observed across our treatment groups is that whether the pictures were enriched, unmanipulated, or depleted dictated their community diversity. So I'm showing a couple different metrics here, breakaway richness being the number of, and this is at the amplicon sequence variant level, so how many unique bacterial species, you could say, are present, so that's richness, and then Shannon's H-index is a composite metric that accounts for both the richness, how many, and how evenly distributed they are in the sample. So pictures that had more mosquito larvae, which is these yellow bars here, they already started with lower species richness, and that effect is amplified later in the season, that relative difference, amplified later in the season from early to late. So this is at day 0 and day 74 in our case. And we see a similar trend that's evident only later in the season in our Shannon's H-index. So especially in terms of that Shannon, both the unmanipulated and enriched pictures are lower than the depleted pictures, which to us is indicative of less bias in the bacterial community composition in those depleted pictures, which supports the idea of some non-random loss or gain, or not gain, non-random loss of in the presence of mosquito larvae of some bacterial taxa, or higher thresholds to successful establishment of newly arriving taxa. And we do find some taxa that are correlated positively with increasing mosquito density, though this is at a quite granular level, and the higher taxonomic levels we're not able to detect those differences. So there's relatively more of ASVs in these broad groups that with increasing mosquito density. And Cryocolas and Actinobacteria, Acetobacteriaceae, acetic acid bacteria, Comomonid is a beta proteobacteria. And there's plenty of literature out there that is implicating some of these groups in insect microbe interactions. So it was encouraging to see this conserved relationship. So Actinobacteria as healthcare for insects is a provocative title that has pointed out this relationship, and acetic acid bacteria in other mosquitoes as well has been observed as potentially symbiotic. Another charismatic example that people may have heard of for Actinobacteria playing a role in insect ecology is in leafcutter ants. So these ants maintain fungal gardens in their large subterranean colonies. And sometimes these gardens are infiltrated by fungal parasites like Escovopsis. And that's where this white patch on the ant comes in. So these ants have specialized crypts that house Actinobacteria in the genus Pseudonocardia that have anti-Escovopsis activity. So it helps keep the fungal gardens parasite-free. Just a cool example of Actinobacteria playing a role in insect ecology. So I promised function also. Let's go through some of the functional relationships we saw between mosquito density and different functional readouts. I'm going to show these data as activity versus total number of larvae along the x-axis that we profiled throughout the experiment, because these are all endpoint measurements. So on the top, there'll be like regressions, on the bottom, box plots. So in the first case, all the way on the left, these are from our Ecolog type assays. So we did Ecolog plate assays, 96 well plates. In every single plate, there's a different carbon substrate in there. And if the bacterial community, if the bacterial community present is able to effectively hydrolyze that carbon substrate, the plate well will turn a different color. So it's a colorimetric assay to measure how fast and how well can the microbes use that carbon source. And we're asking here, is that usage affected by the presence of Wyeomyia? So I'm only showing some of the significant relationships here. So you can look at some others in the paper if you are interested. But hydrolysis of L-phenylalanine we noted because this is an aromatic amino acid that's costly to synthesize. Could play a role in determining what are limiting nutrients in the system. We saw an increase in the propensity of that community to effectively hydrolyze L-phenylalanine in the presence of higher mosquito titers on a regression basis. Kind of consistent with this, we saw a lower efficiency in glycogen. So this is indicative to us to a shift towards the usage of more nitrogen-rich carbon sources like those amino acids. And that maybe this is because mosquitoes are excreting a lot in there. So in the presence of higher mosquito densities, we have higher nitrogen levels, total dissolved nitrogen levels in the system. This is also noteworthy because pitcher plants are nitrogen limited. So this could be a really effective nutrient subsidy that the mosquitoes are providing just because as a byproduct of their excretion. And for dissolved nitrogen, we measured that via another colorimetric type assay. This is like a water quality assay that we used. Lastly, we did microincubations for all of our samples, measuring rates of carbon dioxide production, as indicated by, we had like a pH reactive kind of sandwich 96-fold plate situation, where there was an agar plate on top of the incubation plate where they had carbon substrate, cricket powder on the bottom. And as they evolved carbon dioxide, you have a change in color on the top plate. And we can read that as well as and convert that to a rate of carbon dioxide evolution over time. And we had this parabolic effect. So probably density dependent, as the bacteria are eaten by mosquitoes, you have less activity, but eventually there's high enough provisioning of excreted products to cause a surge in activity again. That's how we think about that. And we measured enzymatic activity with fluorescence assays for protease and chitinase. Only protease had a significant mosquito effect, both in the negative direction. And this is parallel to the temporal effect. So over time, relative to when you're capturing prey, you have less enzyme activity because the microbes inside are hydrolyzing less. So yeah, significant trends for protease in this bivariate representation of the more complex regression we did. A trend in chitinase, but not actually significant. But how we interpret these is that mosquito-induced reduction in these hydrolysis levels isn't negatively impacting the host's ability to acquire nutrients, because the host is like the most efficient player, the host plant, I should say, in acquiring nitrogen. So it's always going to get its pound of flesh, so to speak, by eating nitrogen. So no matter, maybe under extreme conditions, this could be negative for the plant, but under the parameters that we're observing, there was no change in plant growth. And it's probably just bacterial density decreasing. So in our lab assays, we saw bottom-up effects. Bacteria impact mosquitoes. In our field assays, we saw that mosquitoes can do stuff to the microbes too in their natural environment, changing who's there and what that nutrient availability looks like in response, how bulk activity is affected, and how these are modulated in a density-dependent manner relative to mosquito colonization. And there are some correlated taxa as well. We wanted to really hone in on that correlated taxa part in this next part that I'll talk about, and thinking about how these changes could be beneficial and potentially adaptive in this system. So yes, moving beyond correlations to really get at the feedbacks that I promised in my title. So just because we have correlated taxa, are these taxa better for mosquitoes? And is there something about being passaged through conspecifics, being associated with mosquitoes repeatedly over time, that curates a community that is of a higher quality from the perspective of the mosquito? So linking top-down effects to bottom-up effects in this like feedback loop. And we use the eco-evolutionary framework of niche construction to approach this question, which this framework suggests that in addition to heritable traits that go from one generation to the next, there's also lasting modifications to the environment that affect the selective landscape that the next generation experiences. But this is not a framework that's typically extended to microbes in the environment. But we can imagine many scenarios, perhaps in Daphnia or other things, insert your organism of choice here, where organisms may alter their ambient microbial communities in a way that changes the selective pressures they go on to face. Maybe they're better, maybe they're worse as a result of passaging, experiencing the presence of these, of a host species that goes on to acquire microbes from the environment. So is there a sort of external dimension to host-microbe interactions in the cases where hosts have a transient, nonspecific relationship with environmental microbes? So we had a three-tiered approach to think about this question. I'm only going to talk about one of them, which is the gnotobiotic assays here. But we also did isofemale assays where we asked, do females deposit certain eggs, and are those certain bacteria on egg surfaces, and are those bacteria uh, consistent across multiple independent assays of egg laying, and are they beneficial? We also saw if larvae could alter a complex community that was field derived and make it of a higher quality, to see if taxa, once passage through larvae, could be better for larval development. And lastly, we started with, and the one I'll talk about right now is our gnotobiotic assays, where we started with germ-free mosquito larvae, so no microbes, and then selectively reintroduced microbes that had been passage through adults or passage through larvae, to see if they performed better than communities that were only ambient laboratory derived. So is there some benefit to going through mom or through conspecifics for that community in terms of quality for the offspring? Yes. So we generated glycerol stocks, which is just a way of saving bacterial communities for your later use. This included stuff that was field derived. So we had water that had been colonized by mosquitoes or not colonized by mosquitoes in the field. We had oviposition assays, which we just let a single female lay eggs on a sheet under sterile conditions, and then harvested that community, and we pooled a bunch of those together. Then we had a laboratory glycerol stock, which is just their food left out non-sterilely for several days to replicate the ambient microbial community in their rearing environment. And then our conventional positive control, it represents both parental structuring and larval structuring of the community. That's just what's present in the colony by itself, our natural non-sterile colony. So it's our positive. And start with all those glycerol stocks, we exposed azenic larvae to control the densities of those stock communities, and then measured how the bacterial composition was affected in each of those wells, and then also measured the quality of the individuals by measuring how fast they developed, how large they got, etc. All right, and I'm only going to talk about these right now because I won't have time to talk about the others. And so we use 16S rRNA gene profiling again. So these are more relative abundance plots to see what's present in the larval water at the end of our experiment for 5 different treatments I'm showing here. So conventional all the way on the left, parental, that's coming just from mom and dad, those bacterial communities. Laboratory is that just ambient. And then we did different combinations, 2 combinations of the parental and the lab to try to get at the relative importance of laboratory-derived versus parentally derived microbes in structuring fitness outcomes. So we had a 1-to-1 ratio of parent to lab and a 10-to-1 ratio of parent to lab. And something that probably stands out here is orange. And orange is only present in those communities that have had some contact with mosquitoes before, which is pretty exciting to us. So conventional, parental, and in the 10-to-1 condition, we see these actinobacteria again popping up very prominently, but that's completely absent in our laboratory community, which never saw mosquitoes, and when we put our laboratory community at a 1-to-1 ratio with our parental community. Another thing that was notable is that, and maybe it's kind of hard to see, but in the second darkest shade of green, Chromobacterium, that one is also only present in the parental and parental 10 to 1, which we're following up on as a potential bacterial, because that's an easily culturable microbe, easily identifiable. It's purple. It looks cool, and it kills fungi, which we thought was interesting. So that's some sort of other work that we're writing up now. So how does this translate to development? Both in terms of survival on the left here, incubation, and the timeline to development, so how fast they develop, we get levels of development that are comparable to conventional or positive control in our parental and in our 10 to 1 condition. So we get this developmental rescue in the presence of relatively more parental strains, and a rescue also of the speed of development, which is indicated by these density plots. So our laboratory only inoculum takes a lot longer to develop, and actually our 1-to-1 ratio yielded no pupae at all. So they all died before they were pupae, hence there's no accompanying density plot there. All right. So in this series of experiments, we profiled those communities over multiple different types of assays, looking at repeatable colonization by adult microbes, how larvae are modulating the density and composition of microbial communities, and looking to see if preconditioning, we could recapitulate developmental benefits in germ-free larvae of this preconditioning by larvae or adult mosquitoes. And there were conserved taxa in all of these. I didn't have a chance to talk about it all, but Acetobacteriaceae, so acetic acid bacteria, and actinobacteria pop up in all of these different assays that we carried out. Here's a cool graphical abstract that Journey, an undergrad who worked on this project, and I put together. And if you'd like to read more, this work just came out in ISME a few days ago. So I'd encourage you to look into that if niche construction sounds cool to you. And I think I'm going to stop there, actually, because I don't want to go too far into question time, or I want to give people a chance to have questions. But if you want me to talk about how larvae mosquitoes are doing stuff during diapause, we can also talk about that a bit. But yeah, thanks. I'd like to acknowledge my funding sources and the community at Michigan that's already helping me get off the ground and do stuff here. And I'll just place up some other cool stuff that I'm working on but didn't have a chance to talk about today, and I'll take questions. Thanks.
Thank you for that. That was super interesting. One thing I wondered is just, so you talked about how these pictures are very nitrogen poor communities. And then as you have more mosquitoes, they're excreting more and more nitrogen. So where are the mosquitoes getting their nitrogen from?
That's a good question. Yeah, so I wonder if it's in the microeukaryotes and bacteria that they're eating, because that's not like bioavailable to the plant directly. So even though the plant would just like slurp it all up if it was like labile in the environment and dissolved, it's probably not bioavailable to the plant in that form. So the mosquito goes to eat the bacteria or a microeukaryote. It's actually weirdly contentious in the system of like our mosquitoes eating bacteria more or microeukaryotes more, protozoans. Anyways, I think that's where they're getting it from. So they're like transforming that otherwise unavailable, non-labile nitrogen through excretion out. So I think that's where.
Awesome work. So cool. I found it was interesting in that last vignette you showed, if I'm understanding correctly, that the parental inocula led to successful pupation, and the laboratory one did.
Yeah.
But the one-to-one did not.
Yes.
So we saw that like the laboratory was overall not so great. But the fact that on its own it was okay, but when you combined it with the parental, it wasn't. Do you have any idea why that might be?
Negative synergy? Yeah. One thought we have with that is something about microbial interactions themselves. So in other contexts, not in this system, competitive interactions between taxa might cause negative effects in the host, so negative fitness effects in the host. Maybe there's some metabolite or something that's being used in the antagonistic interactions between members of those communities. In our discussion, we sort of waxed poetic about maybe there's more niche overlap between the parental and the lab, because something I didn't show is that we also did these with field inocula. So we put parental taxa in a 1-to-1 or 10-to-1 ratio with stuff from the field, and they didn't fight. Those communities didn't seem to fight. And the parental taxa are some subset of the laboratory environment anyways, because we have lab animals. The perfect experiment would be to go out and capture a bunch of wild Wyeomyia also. We tried to get at that by having picture stocks that were differently visited by Wyeomyia in the field, so with or without previous visitation. But yeah, we don't have an exact composite of that in the field context. So we can't fully get at the niche overlap question per se, but that's our thought of why they might be fighting more. Yeah.
Are there any other questions? Okay, thank you.
Thanks.